This is my project on how the pGLO lab relates to all of AP Bio by Adam McQuaid. So for Unit 1, Chemistry of Life, I chose topic 1.6, nucleic acids, because DNA is a universal code that can be read by all living organisms and is made of nucleotides. And I chose this because the plasmid that held the EGFP gene was transformed into the E. coli bacteria, and that bacteria was able to read that gene because all organisms are able to read DNA because it's a universal code. And for my visual, I chose to draw the central dogma of biology. For Unit 2, Cell Structure and Function, I chose 2.5, Membrane Permeability, because bacteria cells have a phospholipid bilayer membrane that is semi-permeable. And I chose this because the membrane surrounding the E. coli is semi-permeable. So in order to get the plasmid transformed, in the experiment, we used a heat shock to make the membrane more permeable, and the membrane is made of inward-facing fatty acid tails and outward-facing phosphate heads, and as the heat rises, the fatty acid tails become more fluid. For Unit 3, Cellular Energenetics, I chose 3.1, Enzyme Structure, because proteins have a specific substrate that interacts with each other to cause conformational change. And in this experiment, we used an inducible operon, and when lactose was bound to the protein on the operator, it came off of it and enabled RNA polymerase to transcribe the GLO genes that caused the bacteria to glow. For Unit 4, Cell Communication and Cell Cycle, I chose topic 4.6, Cell Cycle, because bacteria are able to reproduce rapidly due to binary fission. And in lane 1, composed of LB agar bacteria with no plasmid and H2O, it served as a positive control, and it resulted in a bacterial lawn because the E. coli was able to use binary fission in the optimal environment to reproduce very rapidly. In Unit 5, Heredity, I chose topic 5.2, Genetic Diversity, because bacteria is able to increase genetic diversity through conjugation, transposition, mutations, transduction, and transformation. In the lab, we used bacterial transformation to allow for the plasmid to enter into the E. coli and have their phenotype be glowing. In Unit 6, Gene Regulation and Expression, I chose topic 6.5, Regulation of Gene Expression, because in bacteria, operons are groups of genes that code for a series of related genes and coordinates regulated. In this lab, we used an inducible operon, and it was used to have the genes, to have the bacteria glow in the presence of lactose, which we saw in lane 4. And for Unit 7, Natural Selection, I chose topic 7.3, Artificial Selection, because humans are able to affect the variation in bacteria through artificial selection. And in this experiment, we were able to use bioengineering to insert the plasmid into the E. coli, which made the E. coli glow when it was introduced with lactose, which is a way that humans are able to affect biology. For Unit 8, Ecology, I chose topic 8.6, Biodiversity, because populations with little diversity are less resilient to change in the environment. And in the experiment, we see the effects of different environmental conditions on E. coli populations. And in lane 2, it can be observed that the E. coli population was drastically affected by the addition of ampicillin, which kills the bacteria. And if the bacteria had a more diverse population, it may have been more resilient to the effects of the environment, and the bacteria that was able to survive from the ampicillin would become the resistant strain. But since we only had one strain of the E. coli in that lane, they all ended up dying.